Our immune system is a complex network of cells and proteins that defend us from harmful invaders. These invaders can be bacteria, viruses, fungi, or parasites. They can also be toxins or even our own cells that have gone rogue. But how does our immune system know what to attack? The answer lies in antigens. Antigens are molecules that can trigger an immune response. They're like red flags that alert our immune system to the presence of something foreign. Antigens can be found on the surface of pathogens or even free-floating in the body. When our immune system encounters an antigen, it mounts a defense. This defense can involve a variety of cells and proteins, but it always starts with recognition. The immune system must first identify the antigen as foreign before it can take action. Once the antigen is recognized, the immune system can launch a targeted attack. This attack can involve a variety of mechanisms, such as the production of antibodies or the activation of killer cells. Our immune system is a powerful force. It is capable of destroying not only foreign invaders, but also our own cells and tissues. This is why it is essential that our immune system can distinguish between self and non-self. Immunologic tolerance is the process by which our immune system learns to tolerate our own antigens. This tolerance is essential for preventing autoimmune diseases, which occur when the immune system attacks the body's tissues. There are two main types of tolerance: central tolerance and peripheral tolerance. Central tolerance occurs during the development of immune cells in the bone marrow and thymus. During this process, cells that recognize self-antigens are eliminated. Peripheral tolerance occurs after immune cells have matured and entered the circulation. This type of tolerance helps to control the activity of self-reactive cells that escaped central tolerance. Antibodies are proteins that play a critical role in the immune system. They are produced by B cells, a type of white blood cell. Antibodies are also known as immunoglobulins. Antibodies are Y-shaped proteins that can bind to antigens. Each antibody has a unique binding site that is specific for a particular antigen. This specificity is what makes antibodies such effective weapons against infection. When an antibody binds to an antigen, it can neutralize the pathogen or toxin. Antibodies can also mark pathogens for destruction by other immune cells. There are 5 main classes of antibodies: IgG, IgA, IgM, IgE, and IgD. Each class of antibody has a different structure and function. IgG is the most abundant type of antibody in the blood. It's also the only type of antibody that can cross the placenta from mother to fetus. This provides passive immunity to the developing fetus. IgG antibodies are produced in large quantities during an immune response. They have a long half-life, which means they can provide long-lasting immunity. IgG antibodies play a role in a variety of immune responses, including neutralizing toxins, opsonizing pathogens, and activating complement. Section 5. IgA, IgM, IgE, IgD, specialized defenders. In addition to IgG, there are 4 other classes of antibodies. IgA is found in mucosal areas such as the lining of the respiratory and digestive tracts. It prevents pathogens from attaching to and invading these surfaces. IgM is the first antibody produced during an immune response. It is very effective at activating complement. IgE is involved in allergic reactions. It binds to mast cells and basophils, causing them to release histamine and other inflammatory mediators. IgD is found on the surface of B cells. Its function is not fully understood. Section 6. Monoclonal antibodies, precision weapons. Monoclonal antibodies, or mAbs, are laboratory-produced antibodies designed to target a specific antigen. They are used in a variety of medical applications, including diagnosis and treatment. For diagnosis, mAbs detect the presence of specific antigens, such as cancer cells or viruses. For treatment, they block the activity of harmful proteins or cells, such as those involved in cancer or autoimmune diseases. mAbs are made by fusing a B cell that produces the desired antibody with a myeloma cell. This creates a hybridoma cell that can produce large quantities of the antibody. mAbs are highly specific and effective, and they have fewer side effects than traditional therapies. Section 7. Antigen-antibody interactions, a delicate dance. The interaction between an antigen and an antibody is highly specific, like a lock and key. The antigen binds to the antibody at a specific region called the epitope. This binding can trigger a number of different effects, including neutralization, agglutination, precipitation, and complement activation. Neutralization occurs when the antibody blocks the antigen from binding to its target cell. Agglutination happens when antibodies clump together multiple antigens, making them easier for phagocytes to engulf. Precipitation causes soluble antigens to precipitate out of solution, making them easier to remove. Finally, complement activation involves the antibody activating the complement system, a cascade of proteins that can destroy pathogens. Section 8. Allergies. The immune system's misfire. Allergies are exaggerated immune responses to harmless substances called allergens. When a person with an allergy encounters an allergen, their body produces IgE antibodies against it. These IgE antibodies bind to mast cells and basophils, causing them to release histamine and other inflammatory mediators. This leads to the symptoms of allergy, such as sneezing, runny nose, itchy eyes, and skin rash. In severe cases, allergies can cause anaphylaxis, a life-threatening allergic reaction. Anaphylaxis can cause swelling of the throat, difficulty breathing, and a drop in blood pressure. Section 9. Conclusion. A balancing act. The immune system is a complex and fascinating system that plays a vital role in our health. Antigens, antibodies, and immunologic tolerance are all essential components of this system. Understanding how these components work together is essential for developing new and improved treatments for a wide range of diseases. From vaccines to cancer therapies to treatments for autoimmune diseases, the field of immunology is constantly evolving. As we continue to learn more about the immune system, we can develop even more effective ways to prevent and treat diseases.